All right, I'm really excited to talk to you all today about some work I've been doing looking at the role of climate and urbanization in driving mosquito preference for human hosts. So most mosquitoes bite a range of animals, but a few have evolved to prefer humans. This makes them dangerous disease vectors. The mosquitoes that bite people spread disease between people. Aedes aegypti is a good example of a species like this. It's spread across the global tropics, and it's a major vector of disease. It's responsible for the Zika outbreaks recently in South America that were associated with these terrible cases of microcephaly. It's also associated with outbreaks of dengue, yellow fever. It's and chikungunya. That have happened in several different regions around the world. But Aedes aegypti originated as a forest-dwelling species in Africa. So in the present day, it's this major vector across the cities around the world, but it came from this very different place. How did this happen? Well, about 5,000 to 10,000 years ago, some populations evolved a preference for human hosts. And you can see that here in sort of our workhorse assay that I'm going to be talking about a lot about here, which is the two-port olfactometer, live host olfactometer. And you can see mosquitoes are in a plastic box, and they have the choice to seek either human odor or the odor of an animal, in this case, a guinea pig. And in this case, these globally invasive populations, in this case, collected from Florida, they're all trying to bite the human host. They're all flying towards the odor of a live human arm. So why did this human biting form evolve? What ecological factors drove the transition? Where and under what conditions did it happen? And what's the genomic basis of this behavioral shift? And for the purposes of this talk, I'm also going to emphasize a little bit what can we expect from this species in the future. To answer these questions, we collected mosquito eggs from across the species' native range in sub-Saharan Africa. We established 50 separate laboratory colonies collected from 27 different locations, and also sequenced 345 unrelated genomes that I'm going to talk about later. These collections spanned a wide range of habitats, ranging from densely populated cities to sparsely inhabited areas, and from wet rainforests with year-round rainfall to the Dry Sahel, where rainfall is concentrated in just a couple months out of the year. We collected eggs, brought them back to the lab, and tested their behavior. Here you can see the behavioral results for those 27 locations. This is from olfactometer data, like you can see in the inset. And the bars show 95% confidence intervals. And so we saw really strong variation in behavior, ranging from strong preference for human hosts, almost as strong as the global invasives, ranging to a strong preference for an animal host. Most populations were animal preferring, but a few showed greater attraction to humans. If we look at a map, you can see that there's a strong sort of geographic structure, with the most human-seeking populations being concentrated in the northwest, in the Sahel region here. Although it's also interesting that you can see sometimes nearby populations had really different behaviors. So Ouagadougou and Ouagadougou in Burkina Faso, very different behavior, even though they're quite close by. Similar dynamics playing out in Senegal. So we wanted to understand that behavioral variation, and so we used ecological modeling approach, incorporating the BioClim2 variable set from the WorldClim data set, and also UN estimates of human population densities. And so after taking into account regional variation, which I'll discuss in a moment, we did see a strong effect of human population density on population preference for human hosts. So mosquitoes that were living near dense populations of humans were more attracted to humans. And this makes sense. They're living in urban environments. They're mostly biting humans in. And so this explained about 20% of behavioral variation. But there was an even bigger climate effect. And we found two variables that went into this climate effect, both of which pertained to the intensity of dry seasons. So one was precipitation seasonality, that is the coefficient of variation of monthly precipitation. This was really strongly associated with the behavior of individual locations. And then the second variable was precipitation in the warmest quarter, which explained a little bit of additional variation after taking into account precipitation seasonality. If we put together these two variables into a single index of dry season intensity, we explain a remarkable 65% of behavioral variation. So what that tells us is climate is actually much more important than human population density in driving preference for human hosts. In particular, we found strong preference for humans in the Dry Sahel. This is sort of a typical picture of what that habitat looks like. It's sparsely vegetated, very dry for many months of the year. And in these areas, mosquito larvae live in human-associated water storage, and this enables them to survive in this otherwise hostile environment for mosquitoes. And so in this case, these are very large, immobile water storage jars. This water is used for cleaning, not really for drinking. And so it's stored there for a long time. It's a little bit dirty. They're rarely emptied because they're big, heavy storage jars. And this is really good mosquito habitat. And so this is where we find really strongly human-preferring mosquitoes. So what's the genomic basis of this behavioral shift? To answer this question, we used 345 unrelated genomes sequenced to 15x coverage as part of the Verily-led 1200 Aedes aegypti genomes project. Overall, we looked at population structure and we saw evidence for three main population clusters. There was a East African cluster, a West African cluster, and a globally invasive human specialist cluster represented by mosquitoes from Thailand and Brazil here. We saw that AIC was minimized at k equals 3, so that was the best fit. But interestingly, when you go to k equals 6, you do see this separate cluster of the most human-preferring populations in Africa. So that's Ngoye, Chez, and Ouagadougou in Senegal and Burkina Faso. They form a separate population cluster, and we think this may represent the human-preferring populations that gave rise to the global invasives, but are sort of differentiated from them because they haven't been through the strong bottleneck associated with spreading around the world. And there was a recent exome sequencing study done in Senegal that also supports this hypothesis. If we look at principal components analysis, we also see that same evidence for three main population clusters, and then we see these human-preferring populations in West Africa extending along the second principal components axis towards global invasives. These are the human-biting populations in Africa. You can see the preference here. If we look at that behavioral variation, we see that it's strongly correlated with the percentage of white scaling on the first abdominal target. For those of you who aren't used to looking at mosquito morphology, that's just the number of white scales in a specific spot on their thorax. This is a character that has been proposed by some people to be associated with the derived human biting form versus the ancestral form. Here we saw that actually continuous variation in this character was related to continuous variation in preference. And correspondingly, we also saw that variation in the levels of ancestry coming from that human specialist cluster, that red cluster here in k equals 3, was related to variation in preference. So the more ancestry they share with human specialists, the more they seek human hosts. We wanted to understand more about the genomic architecture of human preference and specialization on human hosts in general. And so we used the software PCAdapt to see whether we could identify SNPs that were more strongly associated with that human specialist principal components axis, the second PC axis, than would be expected under neutrality. And we saw a lot of outliers distributed across the genome, but they were particularly concentrated in a few key chromosomal regions, and those are highlighted here in gray. And so these regions have particularly high concentrations of outlier SNPs. If we use the population branch statistic, which is essentially polarized FST, we see that the human seeking populations were strongly differentiated from nearby animal biting populations in these outlier loci. And using the FD statistic, which is a measure of shared derived genetic variation, we found that these human biting populations in Africa had increased shared derived genetic variation with human biting mosquitoes outside of Africa. And although I don't have time to show all of the data here, we also saw evidence from analysis of genetic diversity within and between populations, that is DXY and pi analysis, that there's reduced gene flow between human and animal biting populations in these outlier regions, and also that the populations outside of Africa and the populations that seek human hosts and the populations in Africa that seek human hosts are particularly sharing, have less divergence in these regions. And so this is all consistent with a model where preference for humans arose once in Aedes aegypti, likely in areas with intense dry seasons. And genomic changes associated with this shift were concentrated in a few key chromosomal regions. These human specialist populations then spread to urban areas around the world, and gene flow into growing urban areas also drove increased attraction to human hosts in African cities. But human population density and climate are changing rapidly in Africa, so how should we expect Aedes aegypti to respond? We can use our model to start to look at that. So here I've plotted the present day populations across those two ecological axes. On the y-axis, you see human population density. On the x-axis, you see dry season intensity. And in blue, you see the space where these two variables combine to predict attraction to animals. And in red, you see where they would predict attraction to human hosts. The circles represent the present day values, and the arrows represent UN estimates for how these two things are expected to shift by 2050. And although climate was particularly important in explaining present day variation, we don't expect major near-term differences in precipitation that would drive a major change in the aspects of dry season intensity that are important for our model. So although the climate is changing, although climate is important to preference, it's not the most important thing for near-term expected changes in mosquito behavior. Instead, what we see is that the main story is all these arrows pointing up along the y-axis. These are associated with projections of really rapid urban growth in cities across Africa. So a lot of cities in Africa right now are growing really, really quickly. Like in particular, Ouagadougou is one of the fastest growing places on Earth. And it's like a few decades ago, it was not a huge city, and it's growing to a city of millions and millions, like very rapidly. And so what our model predicts is that as these cities become really dense, large urban centers, mosquitoes there may evolve a greater preference for human hosts. This comes with a big caveat though, which is that the projected population densities are larger than the contemporary population densities used to fit the model. So we don't know exactly what will happen as these populations experience new conditions. But the observation that in the present day, invasive populations of Aedes aegypti living in large urban centers across a wide range of climates strongly prefer human hosts is at least consistent with this prediction. So, you know, for example, Southeast Asia is not a terribly seasonal environment. It's quite rainy. The mosquitoes there are really urban adapted and really like human hosts. There's, you know, this isn't a certainty, but it at least makes us think it could happen. And so this is our effort to sort of like predict and monitor contemporary evolution, which I think a lot of us in this seminar are trying to do. Our model makes a actually fairly concrete evolutionary prediction with potential implications for public health. That is, it says that mosquitoes in rapidly growing cities should evolve a greater preference for humans. But it's really important to validate and monitor these predictions. So we're going to ask, do we see ongoing increases in the share of human specialist ancestry and corresponding shifts in behavior or morphological traits? Another way we could ask this question is if we can find archival samples from the past. Do those archival samples show less? So like if we find mosquitoes collected in a city when it was less dense in the past, are they less human specialized? Do we see signs of natural selection at outlier loci? This would be like looking for signs of selective sweeps in particular, especially as cities grow. And this is because this urban growth is such a rapid contemporary phenomenon, we may be actually able to watch natural selection act in real time. And we're going to ask how these changes interact with much more complicated disease transmission dynamics. So although attraction to human hosts is a major component of an overall disease transmission model, you know, mosquitoes biting people spreads disease, disease transmission is a much more complicated process than just that. And so another thing that we're really anxious to find out is, okay, if these shifts happen, do they actually change how disease spreads? Like what can we expect from that? And so these are all sort of ongoing questions that are important for contextualizing and validating these sorts of evolutionary predictions. And with that, I would like to thank all of my collaborators. This was a really large, multinational collaborative effort, and it would have been extremely impossible without everyone working together on this. And so I'm extremely grateful for everyone's work. And I would encourage everyone to also look further into everyone's contributions, because this is a large and ongoing body of work. And with that, I'd be happy to take any questions, and I'm thankful for your time.